Hello, and good to have you all with us. So this is the fourth in this series. Now, that doesn't mean that we've got to the end of COVID. COVID has got a lot more in store for us, I'm sure. But what we do have is we have three outstanding data scientists. So I want them all to come on screen at the moment. Press your screen. There they all are, popping up. Hello, hello. How lovely to have you. So I want to ask you, before we ask each of you to talk to us, I want to ask you what you tell your mums and family what you do to do with COVID. So, and we've got a surfeit of Matts tonight. And I can't call them Matt 1 or Matt 2 because they'd all fight over who was Matt 1. And I can't call them Matthew, one Matthew and the other Matt, because they get worried because you know what it's like. If your mum calls you Matthew and your friends call you Matt, you know you've done something wrong if somebody's calling you Matthew. So we're going to just, so it's Matt S and Matt P. So Matt S, let's start with you. What do you tell your mum you do for COVID?
So I basically would tell her that I work on trying to look at how the virus is spreading across the country and how its genome varies as it spreads. So looking at it in space and time is basically how I would describe myself, really.
Fantastic. And I hope that she tells all her friends. Sam Nichols, what do you say to your mum?
I tell her that the work that I do allows people like Matt S to go off and do their really nice public health analysis. So I build systems and architecture for people to provide data to us, and we pump it out in the right formats to everyone who wants to go off and use it.
And Matt P, what about you? You're on mute, Matt. Oh, there we are.
Yes. So I would tell her that I'm taking data from various different sources. The ends of the pipelines that Sam was talking about, and corral that into categories that can be searched across by members of the public and scientific researchers to find the information they need to add to their own areas of expertise.
Now, I hope you all realise what a rare sighting this is. You've got three data scientists you normally never see. They're part of the infrastructure, but they are absolutely critical. And we're going to hear a bit about what they've been doing and how important they are. They might not say how important they are, by the way, but I'm going to blow their trumpet for them. So we're going to start with Sam Nichols. So Sam.
Hello.
So Sam, tell us a bit more. Tell us what you do and tell us how why we're here tonight.
As Becky was saying in the intro, I work with COG UK. So COG UK is a national network of countless NHS organisations, all for public health agencies, about 20 different academic partners, including Birmingham, which is where I'm currently hosted, the Wellcome Sanger Institute, of course, and these sort of high throughput mega lighthouse laboratories that you might have heard about in the news. And this is all supported by money from the UKRI, which Vivian works with as well. And our job is to, that network produces data. So it takes a sort of sample from a tube. So you might spit in a tube somewhere, it goes to a sequencing laboratory. And out of that, we get the sort of DNA sequence for the coronavirus that is in that sample. And that data comes to us through all sorts of different institutions from across the UK. And my job is taking all of that data, collecting it into some consistent format, and then integrating it into a data set that analysts can go and use for public health analysis.
Thank you. So this trying to get it all in the same format is very important, isn't it?
Yeah, definitely. I mean, I think this is one of the things that COG has done really well and kind of speaks to why we've been able to kind of aggregate so many genomes and make them so available. I think some 25% of the world's coronavirus data has come from the United Kingdom. And this is all because we've presented a simple, unified way for any of these institutes in the UK to provide us the data that we need to be able to kind of put it in our database and make a note of it and be able to find it later.
And without this, we couldn't track variants.
Absolutely. I mean, without this, you might sequence a sample somewhere and that piece of data will just live on somebody's computer or somebody's server in that particular institute. And you'd have to employ people to go around collecting all of that information and trying to build something coherent out of it. So we've really done that at the source. We've provided this central place, this central repository for people to put coronavirus data from around the UK, and it comes to us.
So it all comes to you. You sort it all, you make sure that it's all complete, that it's all in the same standard. Then what do you do with it?
That's right. So once we've decided that it passes QC and it looks sensible and it's not just some random genome that someone's trying to upload to our system, our job then is kind of putting that all in one space, hosting it on a system that we call Climb COVID. And on there, people from around the UK, academics, researchers, public health agency people can come log on to the system and take advantage of the fact that all the data is there ready to be used and do some sort of really cool public analysis. One of the things that people might do is generate phylogenetic trees. And this sort of maps out the relationship of the sequences that you've sequenced. So the idea is that all of the samples that you're looking at are spaced out on this tree, so you can kind of do a sort of put your investigator hat on and try and work out where the virus has come from and where it's going. What changes in it allow us to track it, and that's kind of exactly how we found the variants at the end of last year.
Okay, now I'm going to ask a really stupid question now. So the really stupid question is there, let's say that you've got masses of the Kent variant coming in. How do you spot, amidst all that data coming in, how do you spot the one that's changed and that, oh my goodness me, we've got a different one here?
So the way that we...
Or is it delta or even epsilon?
So we compare everything to sort of the original canonical Wuhan reference. So that was the first ever genome produced from China, and scientists there very generously shared it with the world, which gave us a bit of a head start in knowing what to look for. So we can take sequences that come in from the UK, like I could take one, from today. We can compare it to that, and by looking at how it differs from that reference, we can keep a catalogue of all of those changes, and then you can keep a sort of a database of variants that you've seen before, sorry, mutations that you've seen before that occur together, and that kind of helps you build up a picture of what do we see a lot, what do we see that's expanding, is this potentially new, and this is sort of the basis on how we might look for variants.
Give me some kind of idea of time scale. So I feed my genomes, my viral genomes in. How quickly does it pop up and say, hello, there's one that's different?
Our pipelines run every single day. It starts at 5 o'clock in the morning. So if your sequence and your sample metadata is uploaded to us before that time, it'll be in our data set by the end of the day for public health agencies to take a look at.
Gosh, so that is really quick. And have you sorted them in some way so that it's easier for public health researchers to see what's different?
So there are people who work downstream from us. So they contribute pieces to Climb COVID. One such group is Andrew Rambo's group in the University of Edinburgh. He's got a few people in there, Anya O'Toole and Rachel Colquhoun, who build tools for building these phylogenetic trees, and really those are sort of the early indicators for what looks like a variant. They have their whole Pango lineage assignment system, and they are the people to thank or to blame for the B1.1. Sort of viral identifiers.
So where do our other panelists fit into what you do?
So I guess sort of I'm where the data comes to first, and then, like I said while I was introducing myself, Matt S will probably be the one providing the data to us from the Sanger Institute, and then Matt P, I guess, sort of comes after me. I produce all of this data, we put it out somewhere, and he'll be making sure that it's out on the ENA for people to be able to find. Did
you have the capability to do this before COVID arrived? Or is it something that is, I mean, obviously it's ramped up in terms of the quantities, but was the capability there? How did all of that work out?
The capability was not there at all. We built it in about 3 weeks. So my supervisor, Nick Lohman, sort of said, hey, this COVID stuff seems like it's going to be pretty bad. Maybe we should try and help do something about it. And at the time I was working on LIMS, which is like a fancy way of saying a database for samples. And that was just to keep track of samples in our own lab. And I thought, well, it could be quite useful for keeping track of all COVID samples in the UK. And that is exactly what ended up happening.
And did that involve you? I mean, I've got a vision of you working night and day on this. Was it like that?
It was sort of like a flashback to my undergrad days, finishing assignments at 3 o'clock in the morning, having far too much coffee, listening to strange techno on the radio to get myself in the zone for coding. Yeah, absolutely. A lot of days and nights. And after 3 weeks, we sort of switched it all on, and it worked, and it's worked ever since.
But explain to me then, why is it? I mean, I know that, obviously EBI has a huge reputation in this area as the Sanger, but why is it that we've done so well in this in the UK? Was it something about our existing infrastructure, our existing expertise, the wonderful data scientists we have? Was it, or what was it that apart from having you there, that enabled us to do this so quickly compared to other countries?
So we definitely had the expertise in form of the Arctic network. They were the ones who actually created the sequencing protocol that almost everybody in the UK actually follows to generate the sequences for the samples. And then we had some quite clever, wise prior funding of the CLIME platform. So this is the cloud infrastructure for microbial bioinformatics. This is funded by the Medical Research Council, and it was set up some 5 years ago to provide a place for bioinformaticians like myself to write programs on big data. And so for CLIME COVID, we took a piece of CLIME as it was already running, it was already there. We took a piece of it away, we isolated it for ourselves, and that's where all the data has gone, and that's where all the programs are still running. And I really don't think we would have got things quite off the ground as quickly if we didn't have a resource in the UK like that to just take advantage of.
And do you see this as a resource that will continue? I mean, we've already said, COVID is not going to go away for sure. And we'll see, I suspect we'll get through all of the alphabet and a bit more probably. So how long do you envisage that being there? I mean, is this a kind of, a generation's worth of resource?
I think so. I think we've proven that pandemic's a serious business to begin with. And this may not be the only one in our lifetime. I hope it is, but I think it tells us that we need to be more forward-looking about how we might handle a situation like this in the future. And also there's epidemics around the UK all the time, food-borne outbreaks and influenza. And all of those things can really benefit from sort of mass mobile sequencing like we've seen for COVID. And that all needs to go through some sort of system like the one that we have built. So it might not go through exactly the same one, but we published a paper a couple of months ago about how we built what we built as a sort of notice to other scientists or other technicians in the field to say, this is how we did it. We found that it worked for us. You could go and try it yourself on your own pathogen of interest.
Now, I'm trying to pick up on some of your questions as we go along. And somebody said, and I'm sorry, because I didn't ask this question and I should have done, but what does aggregate mean when you're talking about aggregate data?
I'm so sorry. It's you throw these words around because you're just so comfortable using them. When I say aggregate, I kind of just mean pull everything together, it's just a fancy word for saying that. So
all of it.
Yeah. So the thing that we do when our pipelines run every day, it creates a complete, consistent snapshot of all of the genomes that we've seen in the UK until that particular date. So we sort of aggregate, pull everything together into one snapshot.
So, of course, these are all the genomes of, these are SARS-CoV genomes. These are not the genomes of, some of a couple of questions think that it's human genomes. It's not, it's viral genomes. And interesting question, we know that viral sequences of viral genomes get incorporated into human genomes. Does it happen the other way around? Is there any way that we can trace back the owner or the host of that virus from the viral genome?
Gonna sort of take a little avoidance of that question. It's such a sort of the original origins of SARS-CoV-2 are sort of still up for debate. We believe it's probably come from some animal reservoir somewhere. And yes, if we had good coverage of the genomes of possibly all of those sort of animals that we might think it comes from, we might be able to work it out. But the problem with this sort of thing is it's sort of a search problem, and DNA is only made up of 4 letters. And if you take some sequence of DNA and search against some other genome for that small piece of DNA, you might well find a match that's completely spurious. It just happens to exist in nature. It doesn't mean that it came from that particular host.
Right. So with that's in relation to animals, but humans, we can't tell which human that a particular viral genome has come from. So we can't identify, just to be clear, because some people are asking this, we can't identify the human host in any way from the viral genome that was taken from them.
Absolutely. It's sort of circulating inside of you, completely separate from your genome. The way that when we talk about tracking, sometimes we say genomic surveillance, and surveillance is obviously quite a scary word, but really we're just talking about looking at these viral genomes, which are completely independent from your sort of host personal genome. We don't look at any of that. We don't want any of that. We go to great lengths to make sure that, you know, no one can do that. And really we're looking at how are those pathogen genomes changing over time, and what does that tell us about where it's moving? So of course these samples are linked to very basic metadata about roughly where that sample was collected from, and that's what enables public health agencies to say, oh, we're seeing a spread of this particular variant, or just a breakout in general in this particular area. And that's really useful because, you know, you can go and roll out your vaccine vans to that particular city, for example, and say, hey, everyone, you should get vaccinated. It's a good thing.
So microbes should be very afraid of your surveillance, but humans should simply be grateful because you're showing them where the viruses are and what kind of, in this case, SARS-CoV-2 variants they are.
Yeah.
Okay, terrific. And by the way, the person who said when I said, what do you say to your mums? Was it implying that mums don't understand science? Mums understand everything. Of course they do. But actually, sometimes they do need a bit of help, because sometimes they just say, I don't know what you do, but I think you're wonderful. And actually, those are the kind of mums we're talking about. So thank you very much. We're going to come back to you a bit later. Let's now go to Matt S. Matt S, hello.
Hello. Hello.
So Sam has very nicely set up what he does. Take it on for us.
Sure. So I started in September last year as a senior data scientist at the Wellcome Sanger Institute. So if you're not familiar with the Sanger Institute, it is basically devoted entirely to genome sequencing and has a vast number of resources in order to tackle this. And so we were very well placed at the start of the pandemic. So early on, Sanger turned its attention along with other institutes and the rest of the consortium to sequencing the virus. So we basically work with a lot of the sort of sequencing, with a lot of the sort of testing lighthouse labs. They basically will do the sort of PCR reaction that can tell you if you're positive or negative. And then some of that sort of waste material will end up in a sequencing center, not unlikely to be Sanger, because we do around about half of the UK sequencing capacity. So it could likely end up at Sanger, and then basically that waste material can be sequenced, and then we can try and identify the genome of the particular virus that has infected that particular individual. So
are you, sorry to interrupt there, are you looking at what kind of proportion of these of infections are you looking at? Are you looking at just 5%, 10%, only greater percentage in areas where you think there's a particular penetration of a new variant?
So that really depends. So we partly it depends on how high, what's the sort of overall number of, cases in the UK at a given time. Because if there is a low number of cases, we can try and sequence almost all of them. It's but if there's a high number of cases, we're hitting around about 5-10%. So it's it's basically, well, actually closer to 5-10% is quite a stretch, really. But basically what we're trying to do, if there are high caseloads, we can't get all of them. And you mentioned this idea of sort of, are we looking at particular areas that are sort of targeting this? So we actually, in terms of our output, we actually make a big effort to not show a targeted output. So the reason for this is that we will receive sort of samples that are sort of labeled as particular experiments or particular, targeted, as you said, to particular areas where we believe a variant of concern is prominent. But that doesn't give you a sort of a representative picture of the entire country. So what we try and do is we try and separate these samples into surveillance and non-surveillance samples. And what we present in our output is basically just surveillance samples. And that basically is us trying to get a realistic picture of the spread of genomes across the country without biasing towards particular areas. So that's basically the idea. So in terms of where we actually sit, so I sit in the in the actual sort of product development, which basically sits at the very end of the sort of pipeline. I work with Jeff Barrett, and basically,
when you say product development, I get to interrupt you because I know that data scientists, we think of product development, all us ordinary people. We just think of a product as being like, I don't know, a bottle of shampoo, but that's not what you mean by product, is it?
Absolutely, no. So this is not a product that is sold. This is by product. What I really mean is a package, a package of say some sort of output that serves a particular purpose. In our case, one of our probably our main product at the moment, like I say, not sold, given for free, is our dashboard. And our dashboard gives as live as possible an indication of the genome data that is coming through Sanger. So that data is updated every single week. It is also, there are several checks. I note that somebody asked about the sort of data security aspect, and that is something which is absolutely paramount in everything we do. So in terms of our output, we go through a lot of checks. First of all, we okay what we're doing with public health agencies. What we output is, as Sam talked about, an aggregated output. So we basically provide only information about, which is aggregated firstly over a local authority area. So it's quite large areas of geography, but also over week-long periods of time. And we do that every week. And that basically means that you can't necessarily disentangle a given week's rise in a local authority. You can't pinpoint an exact case. And we also only report cases report areas where there is a certain number of cases as well. So basically we have an output where we try and be as live as possible, but also as secure as possible. So that's
so by aggregating, what you're doing is you're trying to avoid a situation in which people can say, "Ooh, those terrible people in Southall, they've got this condition." I mean, if SARS doesn't have a particular stigma attached to it, but there are a lot of other diseases where there is a stigma, and aggregating allows you to look at a bigger area and not cause particular problems by identifying an area.
Exactly. It means it is basically impossible to. So for instance, if you didn't aggregate, what you may be able to see is, you know that in one particular area, one person went to get a test, and then for a fact that person, if they got pinged, if they tested positive, you'd know what genome they had. That's not what can happen at all with our sort of dashboard. We aggregate over space and time, and also suppress, small number, suppress areas where there are a small number of cases, so that this can't happen. Basically, that is basically. Very much avoiding the idea of people being able to be identified by, providing their data. The data helps us to track this, across the country, but it doesn't. It is highly protected and means that nobody can trace that back to you.
Okay, so the dashboard. Who uses the dashboard principally?
So the dashboard is used a lot by people like one of the sort of major outputs has actually been to you find our data quite often in the news, in papers. So it's used by a lot of data journalists, and you also get a lot of sort of people who are just sort of interested, really and truly. You get people from all walks of life, using our data, because we try and present it in as simplistic way as possible. We also try and move with the times. So obviously, relatively recently, the WHO released their sort of, their new names for variants of concern. So we made sure we went back in and relabel, label on our site which one's which. We also have things like a postcode search, so you can actually look at, okay, so this is like, let me find my area, how's my area looking? And you can try and drill down into that like that. So it's presented as simplistically as, in a way that is easy enough for people to understand and can give people an idea of how the pandemic is progressing across the country and what the sort of makeup of that is.
So you're really acting, I guess, as a translator, in that, there are you, a data specialist, deep in the weeds of your data. But actually, if any of us looked at, who isn't a data specialist, looked at it, we could make head nor tail of it. And actually, then that data would be useless. It wouldn't matter how much data you'd collected. If you didn't present it in a way that was useful to both policymakers and the public and the health profession, you would have failed.
It really is basically trying to make this, as you say, more accessible, because we can publish endless articles about everything we see. Doesn't really matter. It needs to be something that's easily digestible, so that so that people can who are not data minded can get insights from this data. So and it's incredibly important, like you say, for policy. If we can see that there's a particular spike in a particular variant, then those sorts of things may go on a watch list or something, and there may be further actions taken, because if there's a variant that looks particularly deadly, and it looks, well, not deadly, but like that looks worrying, if it's spreading more quickly, then these are things that need to be highlighted. So, it's trying to make this as accessible as possible, but also, and I go back to what I was saying about the separation of sort of surveillance and non-surveillance samples. If we didn't take that out, then you would see quite a biased picture every time that we had an area which that was of slightly more concern. You would see many more samples appearing in that area, because we had targeted sequencing there. And if we had experiments in different care homes, if we're including those data, then that would skew the numbers as well. So doing that sort of filtering as well allows us to give a more balanced picture as well.
What's the likelihood of you missing a new variant of concern? You're only taking 5%. So the numbers that you have of a new variant are very low at the beginning. How do you know that you're capturing new variants?
So in terms of that question, it very much comes down to, so first of all, I say sort of 5%, so, between 5 and 10% is probably a decent number, but it's really and truly, if you imagine that a sort of new variant has to have some sort of dynamics associated with it, it should be growing more quickly than others in a given population. So basically, if we can see, that there is a particular variant growing in a given area, then even with 10%, we are still likely to pick that up. The difficulty is also disentangling that between sort of household effects. So for instance, if you get sort of 5 cases of, and this is, by the way, kind of where the random sampling element of what we do comes in. because if, for instance, if you are sort of sequencing, say if you're getting near 100% of cases, if you're sort of sequencing and you see in a local authority area, oh, there's five of these sequences that are basically identical, this must be a new variant, and we're doomed. Then you go, well, no, they're just from the same house, they're all the same family. So there are these effects to untangle as well. So so it's the thing. It may be that one day we miss it, and then the next day we see it, and then so once you start to see it, it can just sort of grow like that. So it's unlikely that we would miss it forever, but there is that real point that if there are only a few sequences, there is a decent chance we'll miss it at first, and then it will then we'll pick it up at some point. So.
And has this been an exciting time for you?
It has, very much so. So my PhD project was very different to this because it was basically in quite a niche area of structural biology. I have never worked in a job that is so close to policy before. So this is a very exciting job, definitely.
And often in data science, you're a long way from the front line. And actually here you all are on the front line. And you couldn't be closer to the front line. And actually what you do, often in data science, the impact that you have is some years or maybe even decades ahead. And actually what you're doing is having an immediate impact.
Yeah, absolutely. That it is quite an odd thing to see the work you're doing reflected in, in the news, etc. I can give you an example actually around the time of the first the outbreak of the B117 or the Kent variant. I was basically passed a few sort of samples and asked to have a look where they landed across the country. And we sort of plotted them on a map. We could see that, it was mostly down in Kent, but basically everywhere. Didn't actually know anything about this variant at the time. And then a couple of days later, Matt Hancock was in Parliament. So it's quite a strange thing to be this close to, sort of what's going on. So yeah, it's definitely an interesting role. And yes.
We've got some top questions coming in from the audience. So Nick Hindle, a really good question here. Do you use pattern matching to identify variants, or do you have to use more complex algorithms?
So in terms of the lineages or variants that we see, what we basically use is, and Sam alluded to this earlier, so Andrew Rambo's lab in Edinburgh basically defined a nomenclature called pangolin, and this is a dynamic nomenclature based on the sort of phylogenetic tree of COVID-19 variants, of COVID-19 sequences, and we basically use that, and that this basically it sort of works from a sort of pattern recognition. Well, not pattern recognition. It places it on the tree and then uses the tree to identify what label it fits under, and that's actually sort of quite manually curated as well, because the group, Andrew Rambo's group, basically will update their nomenclature if they see like a particular epidemiological event that leads to that needs to lead to the creation of another label. So basically, it's not quite pattern matching, but it's basically placing it on the tree and then seeing which label it should come under using the phylogenetic tree.
Terrific. And we're not calling that pangolin because we definitely know that this came from pangolins. Just pangolins are really nice.
I think I'm not entirely sure of the actual. Apparently Sam was saying they renamed it Pango for that reason, and I think that at the start it may have started off as a bit of tongue-in-cheek, sort of, about pangolins. I'm not entirely sure why they called it that, but
right.
It's
very good. Okay, so we're going to move on to Matt Pearce now. So thank you very much, Matt S. Very good to hear all of that. Matt Pearce, hello.
Hi.
So where do you fit in all of this? We've heard about the dashboard, we've heard about the person who feeds it in. What's your part in this?
So I am the technical project lead for EBI Search, which is part of the EBI's web production team. And what that is basically a search engine allowing people to search across many of the EBI's data sets. Back in, I think, March 2020 or April 2020, obviously when the first lockdown was going underway, the team were asked to pull together a portal gathering the data from across the EBI's data sets and bringing it together under a single COVID-19 specific site that would allow access to search across all of that data.
Why was there a danger then that there would be kind of puddles of data in various places and somehow things would get missed because not all of the data was being searched together?
Yes, that was a definite danger. Several of the teams across the EBI have their own APIs for searching across their data set.
So APIs?
What does API stand for?
It's a kind of handshake, isn't it? Between,
yes.
Between different systems.
Yes. An application programmable interface, I think. But yeah, they had their own means of accessing this information. But pulling that together into a single portal would have taken months. EBI Search is in a fairly unique position where we have metadata from across those data sets, and making it available is relatively straightforward. We have our own API and our own access to those data sets. So the COVID portal provides either some of the data sets that we've got are curated specifically for the portal, while others use filtering to limit the results from the various EBI data to narrow it down to COVID specific data.
Now, Sam was talking earlier about standardizing. And I know that somebody in the audience has said, what exactly do you mean by standardizing? But here you have a real example that you have lots of different data sitting in lots of different places. How do you make it have the same, make sure that you're comparing apples with oranges, apples with apples rather than apples with something else?
Sure. We do a lot of data massaging. We also keep our data sets separate and provide a unified search across them. So if you type something into the search box, it actually goes away and searches a dozen, or in the case of the EBI search front end, around 100 actual data sets in parallel to bring back the results that you can then drill down into if you're interested in viral sequences or host sequences or literature or so on.
And again, was this something that you had to work through the night in order to develop all this very quickly?
Yes, I believe the COVID portal was launched across the space of about 2 weeks. We had a number of members seconded from teams across the EBI, user interface people, scientists, data specialists, and so on, and also obviously the EBI search team providing the search backbone for the site itself.
Now, what would have been the implications if scientists hadn't been able to use this kind of search engine?
It would have been a lot slower for them to gather the information, basically. We hope that we provided a system for searching across this data quickly, making it possible to drill down into the data We're using, standard search tools such as faceting, sorting. We've also got a particularly powerful feature in EBI search called, which we call cross-referencing, which basically links data from various data sets together. So if you find a viral sequence that you're interested in, you can then click through to that and see that you've got references in, say, literature. There might be references in the imaging sections of our data, which, while not directly linked to the thing that you're looking at, reference it in a kind of sideways way. It allows you to, step sideways and look into other areas where that record has been referenced.
Now, I said I was going to blow all your data scientist trumpets for you, but actually what you've done is you've made things immeasurably easier for scientists who are researching this. So therefore, you've done something directly to impact COVID. And how did that make you feel?
It feels great. Being able to contribute to the wider community is, it's a real driving force for me personally. And I know that the other members of the team feel the same way. Our aim is to make this data open and accessible to as many people as possible, whether they're researchers or whether they're, regular members of the public. So it's a good feeling.
Now, we've got a good question for you from the audience. The problem of making diverse databases interoperable has been solved in many different contexts. Do you learn from industry best practice, or do you start from scratch?
A bit of both. My background is basically as a search consultant. I spent a lot of time up until January this year kind of working across various industries. So what we're trying to do is adopt the best practices. The advantage of the EBI search is that because we are using different data sets, we can be very flexible in what we make searchable. So the various fields for sequences are not going to be the same things that you're going to want to search over for literature, for example. So we have the flexibility, but we also, we've provided a RESTful API, sticking with, the best practices across industry. Anyone can access this data through that API. You just call a URL, you pass in your query, and whatever filtering you want to apply to it, and you get a set of results back in XML or JSON or a range of formats.
Now, those of you who are not from the data world, you will have had a new term there. You're a search consultant, and that's what the job you did. And I bet you, if you said you're a search consultant, people would have not a clue what you did, but actually it's a really important thing, because you enable us to find our way through the thicket of information that is now everywhere in data sets.
Yeah, exactly. I spent 8 years trying to explain to people that search wasn't just Google.
Other searches are available,
yes,
including
exactly.
that. Fantastic. Okay, what I'm going to do is I'm going to invite all our speakers back again. So if you'd all pop your cameras on again, because I wanted to ask all of you some questions about this. I mean, one of the things is that we've all seen sequencing on the front pages of newspapers, and actually science has had an extraordinary moment during this pandemic, because people can see, with their own eyes, how it's being applied. And is the same true of data? Sam?
Yeah, I think so. I mean, the work that I've been doing, I started in March of last year, but it wasn't really until December when we kind of discovered the alpha variant, and the sequencing project itself really ramped up. That's when really everything hit the news, like just before Christmas. And like many people, I didn't go back to my family that year, but they were sort of like, well, how's your year been? What have you been up to? And I'm like, well, if you turn on the news, they're talking about the project that I work on. And, you know, kind of what Matt S was saying earlier, it's just, you know, we've been working as scientists on things that seem quite abstract, like sequencing genomes. Oh, that's so cool. Why is that useful? But really now it's kind of proven itself, and everyone knows about sequencing as a technology and why it might be useful. So it's quite exciting to be a scientist in a field that's kind of hit the public eye, so to speak.
Matt S.
Yeah, so I think that it's very interesting, you know, how, like you say, how it's working in something so relevant has been, you know, quite a strange thing. Like I say, I worked in a very obscure PhD on a very obscure PhD project. If anybody watching this has read my paper, I'd be absolutely shocked. But you know, working on something that is relatively high profile has been pretty great. Yeah, I think the fact that the public is, you know, becoming more, you know, it has as well forced the public to become a bit more sort of, you know, happy about looking at data. And I'd say, you know, obviously that is something which I think a lot of people have really improved. I do as well, though, think that there are, and I'm not going to name the particular newspaper I'm talking about here, but one of the ones who has, you know, before shared our data. So for instance, at the start of the B16172 outbreak, the Delta variant outbreak, this particular newspaper put a big, scary looking map of the UK and put a local authority and said that it was 100% Delta. And they said that because it had one sequence that we'd sequenced in that local authority area. So you have, whilst I'd say the public has got, you know, definitely got better in terms of data, partly because, you know, because it's become part of our lives now. I think that there are some publications that maybe want to sort of have a bit more of a look at themselves and what they're doing with that data as well.
Yeah, there's a way to go yet to try and explain data. Somebody also is asking about the risk of hacking attacks. Matt, can you help us with that, about hacking? Matt P.
Because I can't.
Oh, no, okay. Oh, sorry, Matt S. Sorry, Matt S. Hacking.
Oh, well, I have to say, first of all, it's not really my area. But in terms of the, so we have, you know, Sanger has a very well, I know, I can only speak for Sanger, and I know has very well furnished IT security. We have very rigorous processes in place. I don't really know how those processes work, but I can tell you that, you know, in terms of the data security, we have a lot of it. And because we are contracted to the Department of Health as well, they stipulate a certain amount of security that we have to have for our data sets as well. So in terms of, and we also have very strict rules as to what we are able to do with this data. So, you know, we can only look at it in a certain place. We cannot, for instance, move any of it onto our personal machines that, you know, where it can only be stored on basically servers that are on campus all the time. So basically we have very stringent protocols in place for handling this data that we can't, we have to adhere to, and for very good reason.
So on the one side, this is, you know, you have to have highly secure environments, but on the other hand, you have to make it as open access as possible. So you have to run both together, which may seem strange, but, you know, again, we go back to this thing about your data has to mean something to people, and it has to be translated, and it has to be accessible, so as many people as possible can use it.
The way to do that is a little bit strange, because it basically means sharing your code, but nobody else can run it. So it's pretty much what it means. You can't give other people access to your data so they can run your code, but you can tell them what your code is.
Now, it will not have escaped your notice that all our panelists are male tonight. That wasn't intended at all. We've had these some sterling efforts by these panelists here who've come in at the last minute because our intended panel, not that they weren't our first choice, you understand, but dropped out at the last minute. So normally these panels would be diverse, and the fact that they aren't tonight is a reflection of holidays, pinging, and general disasters. So but I did want to make the point that actually data isn't always neutral, and diversity is a really important point in data collection. How do we know that we're collecting the right sort of data amongst a sufficiently diverse population? Who wants to take that? Sam?
Yeah, so I guess this is more of a question for someone who isn't me, conveniently. It's more of a policy sort of thing. People in public health agencies will kind of decide where the sequencing should be applied. They are in close contact, certainly for England, with the Office for National Statistics, and they kind of decide where they're going to kind of apply more sequencing to ensure, because they know what the demographics are and we don't, and they'll ensure that we're getting the right diversity of sequences from underrepresented areas, minorities, and all of those sort of important aspects of how socioeconomic impacts of how will impact the pandemic quite dearly. And I think we're really still trying to grasp with that data, because it's sometimes quite difficult to make available.
This data scientists do tend towards the male. It's quite, there are not enough women yet in data science. Does actually that have an impact on how you all work and the things that you think are important? Matt, I'm going to go to you on that one. Matt P, sorry.
Speaking as a software developer and software engineer, I'm pleased to say that I've come across far more diverse colleagues over the last 10 years or so than previously in my career. I've certainly worked with plenty of women coming through into STEM careers. I have colleagues from all over the world. I am, I think, one of only 2 British people working within the web production team at EMBL EBI. My colleagues are from around Europe and across the globe. So that's kind of how I see the importance of diversity in computing and in software, encouraging people to get involved, making sure that nobody is excluded as well. There can be, certainly in earlier generations of computer science, there was, a level of gatekeeping, which has kept people away. It's put people off for various reasons. The kind of machismo and braggadocio that goes around with, certainly software development communities has had to be adjusted. And those, people called out for their behavior and their language.
Okay, so let's, you've become poster boys, I'm going to say, for data science. If there are people listening to this who perhaps are students, perhaps who got their A-level results only this week, how would they become somebody like you in academic terms? Matt S, how would they end up doing what you doing it? I mean, how did you end up doing it?
Well, if I was to put it into 2 words, first of all, I would say learn Python. Basically, learn a programming language. It doesn't really matter, well, actually, it does. Well, basically, it doesn't really matter what you already do. Learn a programming language, and then you can basically, if you can learn a programming language, figure out the sort of principles around how you want to apply it along the way, because pretty much the major thing is learning the fundamentals.
So learn the fundamentals, and the world is your oyster. Sam, how about you? How did you get into this?
So my background is in computer science and statistics, but I've been coding from a very young age because I was lucky enough to have a grandfather who could build me a computer from scrap parts. And so really I've been coding for almost my whole life. And I completely fell into biology by accident by replying to an advert for an internship at the Sanger Institute. Didn't know what it was, who they were, what they did. And that summer was really quite important to me, because it was like, oh, wow, I have these computing skills, but I can apply them to something more, not just building a website or making a game or something. I felt like I was doing something, more important with those skills. So yeah, as Matt S said, I definitely encourage people to learn a programming language. And I think the nice thing about kind of where our industry, where this field of software engineering and science is heading, is so much more democratized. You can just Google, how can I learn Python? And there's going to be a million different websites that will say, here's a Python interpreter. You can type Python into it. It'll tell you what you're doing right, what you're doing wrong. You don't have to even go to university anymore if you don't want to. You don't even need an A level. You can definitely teach yourself a lot of this stuff at home. And I think certainly for sort of really heavy programming jobs, it's really what about your sort of software portfolio you can show. Like, what have you built in the past that's really cool? What excites you? And I think that matters a lot more than the grades. And I think that's really where our industry is heading, and especially in science as well, because I think science is coming to a bit of a point at which they're saying, hey, we haven't really been funding software people. We've been funding the sort of the people at the bench and all of that sort of thing. And I think really there's going to be a big shift over to supporting a new generation of, I guess we call them research software engineers, RSEs. And I'm looking forward to seeing that.
Research software engineers, Matt P., is that something that you're going to be looking forward to?
Yes, absolutely. It's.
I mean, how did you end up where you are?
Much like Sam, I grew up around computers. I was a kid in the 80s during the microcomputer boom, and I always wanted to be a programmer. I went to university to learn how to be a programmer. I spent much of my career kind of wrangling data in one sense or another. I worked in the media for quite some time as a software engineer, and then ended up as a search consultant. And among that, I was working. We spent 18 months. The company that I was working for at the time, we spent 18 months working with the EBI on the BioSolar project, which was an attempt to expand and make bioinformatics search more useful to the community by using open source search tools. And from there, I saw an advert on LinkedIn and applied for a job at the EBI, and here I am.
Fantastic. So I know that data scientists are now in huge demand everywhere. So if you're a parent and you're listening, if you want your child to keep you in your old age, can I recommend that you suggest either bioinformatics or data scientists, because they are in extraordinary short supply. But I was wondering whether all this extraordinary work that you've done that's really become much more visible, not just to the public, but actually to funders. I mean, you've really proved your value. Does that mean, do you think, that there will be more funding? And this is a question that came from the audience from Nick Kendall. Will there be more funding for genome sequencing and related data science? Matt S., how about you? What do you think?
Well, I can actually sort of attest that that is sort of already happening. So as part of my role at Sanger, I also work on the Malaria Gen project, which is basically a malaria genomic surveillance project at the Sanger Institute. Well, I say at the Sanger Institute because it is partly the Sanger Institute, but it's also a collaboration with other institutes across Africa and Asia, etc. as well. Malaria has been doing genomic surveillance for years, but it hasn't had the profile that. So partly because genomic surveillance is not mainly focused on publishing papers, it is focused on delivering insights and delivering those insights to policymakers. So, and there is definitely more interest in terms of the in terms of the malaria work that we do now. So in terms of the genomic surveillance, I would say that is already happening, and it's a great thing, because it's a really important thing to be able to have an understanding of how a particular disease is changing over time and space, and genomics gives you pretty brilliant way to do that. So I think there is going to be more funding, I think, and I think it's a great thing.
And Sam, you're in a university. Are you seeing universities getting a bigger share of the research pie in terms of data science?
I think for people like me, like private single academics, maybe looking to get a fellowship, that money is sort of coming at a much slower rate. I think it's easy to fund very big successful projects and say, yes, this sequencing COVID's very important, sequencing a new pathogen is very important, but a lot of the stuff underlying that is people like me, people like the two Matts, building new novel software, new ways of doing things, new ways of storing data. And that really nitty gritty computer science is kind of not really exciting. And I think it's taking a while. I think we're coming to it now, in that funders are really seeing that this stuff's really important. And kind of from my personal experience, I've always felt torn between having to make applications to the Biological Research Council and the Engineering Research Council, and the engineers say, oh, this is too biology for us, and the biologists say this is too technical for us, try the other guys. And I think we're really looking at seeing a kind of more meshed sort of research council approach. I think people are really looking at how can they fund people across those borders and fund things like what we do. So hopefully that will start happening for people like me.
And indeed, that was exactly why UKRI was set up, or one of the reasons was because of this interdisciplinarity that, and now almost every project, I suspect, in the medical field has a data scientist attached to it somewhere or other. And you're also seeing, very interesting, I think, that, in medical schools, programming is also considered part of the curriculum. It's no longer the case that you're allowed in medicine to say, oh, I don't do the programming thing. I mean, you might not do very good programming, but you're expected to have a basic knowledge. And I think that's, as we go beyond COVID, we'll see more and more of that insistence that people have these kind of skills as a basic. Another thought, the availability of data. I mean, we're getting more and more data flooding towards us. When we have these vast amounts of data, doesn't that actually make your jobs much more difficult? But let's start with you, Sam, on that one.
Yeah, absolutely. I mean, I think no one had any idea what the trajectory of the pandemic was going to be when it hit the UK last year. And the software that I was building had kind of 25,000, 50,000 samples in mind. We thought that was quite a big number at the time. And, we hit that within a couple of months. And by the end of last year, I think we'd hit nearly 500,000 sequences. That it was basically an exponential growth. And that led to some real problems in the software, because suddenly things that were taking 5 minutes yesterday are taking 10 minutes today and 20 minutes tomorrow. And so my job was kind of spinning all these plates in the background, keeping an eye on how long things were running and what things were doing and what computational resources they were taking up while they were doing it. And then also actively trying to work out how to reduce the impact that the level of data was having on our systems. And I think, touch wood and touch my desk here, that we're going to approach 1 million samples in maybe a month or two of time, and everything's holding up at the moment. But there's really constant work going on behind the scenes just to keep the things that run every day that look like they're absolutely fine running every day. It's a huge amount of work just to keep those things going, let alone do new analysis and new techniques and all of those sorts of things as well. So there's a lot of innovation just keeping the stuff that we already have going.
And Matt P, you as a search consultant and looking at searching, the more and more data you have, presumably the more difficult that is to do. But also all of this, high input computational work is not carbon neutral, is it? I mean, we have to really think of these things, when you have huge amounts of data and you need to do all this searching and use all these very high powered computing to do it. Actually, there is a significant environmental impact, isn't there?
Yes, there is, because apart from anything, data centers, they use vast amounts of electricity keeping disks moving, but they also use quite a lot keeping things cool. It's not something that is kind of that widely known outside of the computing world, but obviously computers need to be kept cool, and they start breaking if they get too hot. So air conditioning costs at data centers are a major issue. The EBI is actually in the process of opening a new data center in Harlow, which is aiming to be, if not carbon neutral, then significantly more environmentally friendly by using some revolutionary cooling techniques. Basically, I can't actually pretend to know what they are, but it is...
It's very cold.
Yes.
It's very cold. So, Mattas, we're coming towards the end of our session. As if there are any last questions that you want to put in the audience, please put them in now. But Mattas, one particular thing that has come in from the audience is, are other countries using similar systems to ours? And do you go out and advise them? Do they look to the UK for advice?
In terms of other countries, I would say that the US has massively ramped up its sequencing capacity over the course of the pandemic. Especially, they went from, if you can actually, there's a very good plot of basically the number of genomes sequenced by the UK and the US just suddenly going up through the ceiling. So the US is one that massively scaled up. Another one that was already very established. This is Denmark. So Denmark already had very good sequencing coverage, and they were quite quickly able to roll out sort of similar levels of surveillance at similar levels of granularity. But in terms of the sort of capacity at the start of the pandemic, very few countries could really rival the capacity of the UK. So we were the only country that was able to deliver a surveillance system that could be as granular as a local authority area. So basically, we do very much, we do have something to be proud of in that, very much so. And you do get, so I can't really, we do have there is a partnership that's the Sanger has opened up with South America, with some South America countries. There is basically helping to set up some sequencing capacity in South America as well. So yes, absolutely, people are sort of turning to the UK, the UK institutions for help. And Sam has helpfully put here that we have apparently had the second biggest system within 2 weeks behind China. So I can't take credit for that though, Sam. That was very much Sam's.
So one final question, which is for Sam, which is about, and I'm not sure whether this is a data science question, but whether what you do can spot variants that are either more infectious or, on the other hand, more virulent.
This is the sort of analysis that we can enable with the infrastructure that we've built, but it really takes going back to the hospitals in question, and doing the reason it takes so long to find out these sorts of things is because we can't just say, oh wait, it's much more dangerous, it makes you much more ill based on 2 cases. You kind of have to really gather the evidence, and that's sort of done outside of COG, outside of our sort of sequencing effort. And the thing that we help with is that when samples come to us, they're labeled with an identifier, and the hospital that actually sent them knows that identifier and can query our systems to find out what genomes that have been sequenced for them. And they can use that information to go back and say, this genome looks interesting. It looks like it might be related to this one on this ward, or from someone who's traveled to this country. And I think the infrastructure is what allows that to happen, but really it does involve going all the way back to the point of care, or where the sample was taken, or the local public health agency. There's so many people involved in answering a question like that. PHE does produce technical reports, I think every week or every other week, that kind of goes into how their data analysis supports things like this. So they might say, for a particular variant, we have moderate confidence that it's more virulent or more transmissible, but it takes a long time. Even we saw this even with Delta. You kind of have to see it happen to be able to say, yes, this is what's happening. So we wouldn't necessarily know from the off, from its genome, that it's much more transmissible or much more virulent. You've kind of got to see what happens out in the environment, because that's kind of where the virus is, that's what it's mutating, that's where it's getting its advantages or disadvantages from.
Well, I want to thank all of you, the 2 Matts and Sam, for being with us this evening, because we've had a real glimpse behind the curtain, I think. These are people who you really don't see very often, but who, as I hope you've now realised, are absolutely critical to our response to COVID, and indeed many other things besides. I hope you've also learned that if you are just got your A-level results this week, that data science is a top thing for you to do, and that the world will be your oyster, and that you can, if you get data science, if you get involved in computer science and use your programming skills, you can do almost anything and make a huge difference. I want to thank all the audience for sticking with us. Most of you who joined have all stayed, so thank you very much for that vote of confidence. And thank you to Connecting Science for this Connecting COVID event. As I said, it's the last in the series, but ladies and gentlemen, this is not the last of COVID. We'll no doubt see you all again soon. Thank you to our panel. Thank you all to you. Good night.
Wonderful. Thank you so much, Vivienne. I was just going to come back at the end here just to close out and say a huge thank you from Wellcome Connecting Science to you as chair, and to all of our panel for such a lovely conversation, taking us through the endlessly delvable world of data. There's a huge amount to see with that. We had lots of really interesting questions coming in from the audience, so thank you so much to all of you for submitting them. Hopefully we got through all of the things that you've been thinking about, the really interesting themes that came up. I'm just here to close out with a few bits of our housekeeping, to say that at the end of this, there'll be a link posted to our event survey. I know everyone asks these of you, but if you have a few minutes to stay on the line and fill that out, it's really useful for us to think about how we plan our future events, particularly, as Vivienne says, we draw this one to a close. We'll put some links in the chat to the work of our chair and our panel for the evening, so you can follow up with those. If you haven't seen the previous 3 of the COVID connections, if you are able to join us, that's fantastic. Thank you for coming back. If this is your first one, then you're able to see them on demand on our website. So if you follow the link in the chat to those, you'll get them. So it leaves me just to say thank you all again for being here. Enjoy the rest of your evening. Thank you to Vivienne, Matt and Sam, and we'll take care, and we hope to see you again before too long. Thank you.